cancer that originated in the 1920s, and something new because none of what I'm going to tell you is published, and it's really all been done in a very small group over the last 9 months to a year. So essentially, the mitochondrial theory of aging really originated with Warburg, and he believed that mitochondrial damage, or accumulated mitochondrial damage, led on to cancer. And this has really held sway and really got lost in molecular biology in recent years, but it's still there. And the fact that many glycolytic tumours, cancers are glycolytic is part of what we recognise now. Glycolytic cancers are often resistant to chemotherapy. They tend to be more invasive, more metastatic, and they take up more glucose. So this is really the basis of PET scanning for cancer that's used in a wide number of solid tumours. Right. So some ideas about metabolic flexibility in cancer, because I think this is quite important. Proliferating cells have been shown in the 70s and more recent evidence to be largely glycolytic, or at least to switch unbiased towards glycolytic metabolism. And you can imagine that this might be to protect their genome, less reactive oxygen species in the mitochondria, less DNA damage, both in the mitochondria and in the nucleus, and perhaps greater survival benefit. Non-dividing cells, our muscles and our brains, use energy largely through mitochondria, and of course it doesn't matter with these cells if you get DNA damage. We know that hypoxia imposes stress on a cell, and that this stress biases the cell towards glycolytic metabolism. And this is a characteristic of many cancers and growing solid tumours. They increasingly become glycolytic because of the hypoxia. And there's also the issue of nutrient stress and altered metabolism because of nutrient stress. Nutrients just don't get into the tumour efficiently because of vascularization issues, and this seems to be a characteristic of tumours as well. And the question really we wanted to answer was whether or not this can be demonstrated in a tangible sort of a way, whether cancer stem cells were actually glycolytic, and this might be one of the reasons why they survive, a bit like normal stem cells. And so we were interested in imposing exclusively glycolytic metabolism on tumour cells. So we used the mitochondrial gene knockout model to model tumorigenesis with the B16 melanoma model. That imposes a pure glycolytic metabolism on these cells. Now, you'll maybe all know that we have two genomes, one in the nucleus and one in the mitochondria. Or you might not realise that knocking out the mitochondrial genome doesn't kill the cell. The cell, in fact, can survive. And this is, of course, the survival advantage that goes back to the ideas of Warburg in the 1920s. So Rho zero cells then can be generated quite easily, certainly in tumours. It hasn't yet been done in somatic cells. They survive under hypoxic conditions, in fact know a little better under hypoxic conditions. They're resistant to a variety of drugs, anti-cancer drugs, and they have increased glycolysis, and they have increased reduced protein nucleotide. And this is perhaps what you might expect in this sort of metabolism that is very biased towards generating ATP through glycolysis. So we generated from this metastatic melanoma cell a Rho zero model. We showed it was It did not contain cytochrome B subunit that was encoded in the mitochondrial genome. We looked at the growth characteristics of these cells, and the row 0 cells grew. They survive indefinitely in culture, and they grow a little slower, usually at about half the rate of normal cells. Interestingly, the row 0 cells grew a little bit better under hypoxic conditions. So the study design then was to inject these row 0 and control cells into mice, both intravenously, where they would grow subcutaneously, locally, and also intravenously, where they would then seed in an organ. And it's been well demonstrated that the primary organ of metastasis with this tumour is the lung. So what we saw was, in fact, delayed tumour growth. Cells without any mitochondrial DNA or electron transport function are unable to produce ATP in the mitochondria, are still able to grow. They take some time to adapt to the mouse environment, about 20 days, but they grow, and they grow, as we would expect, at about half the rate of the wild-type cells. So knocking out the mitochondrial DNA doesn't compromise tumorigenicity per se. What happens when we inject intravenously? Well, surprisingly to us, and it's perhaps exactly the opposite to what we expected, these cells did not form lung tumours. So taking out 55 days, no lung tumours, and in this model you can detect black melanic dots in the lung, probably within 5 or 10 days. So these row 0 cells had in some way developed resistance to